Thank you so much, Rodrigo, for inviting me. So what I'm going to talk about is a little bit less grand scale than what Dr. Lord talked about just now. So we're also doing some kind of digital agriculture, but on a much smaller scale than what was presented in the last presentation. And we focus mainly on analysis of omics data in fields. So traditionally, where I come from, which is the systems biology field, what people do is they grow different plants in a lab setting in a number of replicates, and then they apply controlled perturbations, either genotype perturbations or environmental perturbations, on these plants in the lab, profile a bunch of omics layers and some phenotypes, and then try to link all of them together. One of the issues here is that whatever you get from these lab data is often not really representative of what happens in the field setting. On the other hand, this traditional systems biology approach to apply it in the field is pretty difficult, because you need to be able to do a huge number of controlled perturbations in a field setting, which is not evident. So, but we thought, well, actually, from a systems biology perspective, any perturbation is a good perturbation. So why don't we just use the naturally occurring microenvironmental perturbations that happen in the field all the time? So we canceled out the genotype factor. So we're looking at individual plants of the same line, which are virtually genetically identical. And we profile all of them individually in a field setting. And we generate omics data layers on them. We phenotype all of them individually. And then we try to figure out whether there's information in these coming out of these natural microenvironmental variations that tells us something about how the gene networks in these plants are structured and how these networks impact individual plant phenotypes. That's the main idea. And we've tested this previously on two different crops. This is a picture from a proof of concept study in maize, which was published a couple of years ago in Molecular Systems Biology, where we essentially profiled transcriptome and metabolome for a number of individual field-grown maize plants. And we used this to predict gene functions on the one hand, and on the other hand, to try and predict some phenotypes of these individual maize plants. And essentially, we could show that the data that we generate is not just random noise, but that there's really biological information in these individual plant omics profiles. So for the sake of time, I'm not going to go in a lot of detail on this particular study. It's published. Same for this one. We performed a field trial. Well, most of you would not really call this a field trial. It's a 5 by 5 meter plot of Brassica napus plants, so 100 plants in total. We couldn't really go a lot beyond that because omics data is still pretty expensive to generate, right? So we essentially profiled 62 of the 100 plants standing in this field. And again, over here, we could show that what we get out of this in terms of prediction potential and omics data is not pure random noise. We can do some predictions. The people involved here were two brilliant PhD students in my lab, Sam and Daniel, and this was in collaboration with ILFO, so the Flemish Institute for Agriculture, Fisheries and Food. So what I'm mostly going to talk about is the follow-up field trial again that we did on Brassica napus. So this is a little bit of a bigger one, but still it's only 10 by 10 meters, 484 plants. People working on this mostly in my lab are Michael and Steen. And so essentially what we did was sow a bunch of plants in a field at ILFO. We took soil samples for soil microbiome profiling for every plant. We also did nutrient profiling for every plant from these soil samples. Then we followed up the rosette areas in the fall, just by not by drones or any robotics or something, just by manual picturing of these plants in the field. and then applying some convolutional neural network to get the information out. And then in autumn, we harvested one leaf for every plant, then phenotype these leaves as well, and also use samples of these leaves for RNA sequencing. So we have for every plant one time point of RNA sequencing in the fall. The rest of the plants were still standing in the field, so they continue to grow. And then at the end of the season, we harvested all of them and also generated some other microbiome layers for roots, soil, and seeds, and then also did some final plant phenotyping. And then we tried to link these omics profiles in the fall to all of the phenotypes and mainly the final phenotypes in the spring. So the first thing we did on these data was multi-omics factor analysis, so with a tool called Mephisto. One of the advantages of Mephisto is that it's able to also take into account spatial context. So obviously we're growing in a field, there's spatial autocorrelation effects here in the field. So the factors that we get from here will take into account this spatial autocorrelation. For instance, you see that factor 2 here is a factor which is kind of oriented top to bottom in the field. And then if you look at the loadings for these factors, this mostly explains variance for the nutrients and for the sensors that were in the field here, which makes sense. So some of these are pretty highly spatially autocorrelated. For instance, if you look at manganese, in this little field, there's a gradient from top to bottom. For ammonia, there's like more at the top here. For lead, there's a gradient from left to right. For phosphorus, there's a gradient from the top left to the bottom right, and so on. And so there's different kinds of variation in the field. Actually, this was surprising to us. This is a really small field plot, but you still see a lot of variation really on a very, very low level. And then the sensor data as well. So average soil temperature, as you might expect, varies from south to north. The relative humidity was, for instance, higher in the middle of the field than at the edges, and so on. So returning back to this multi-omics factor analysis, the factor explaining most of the variation is factor 1 here. And then this mostly explains variation for the transcriptome and the phenotypes, and less so for the other data layers. So we can already anticipate from this that the transcriptomes that we generated in the fall will be most predictive of the phenotypes that we get in spring. Some of the other data layers, like the leaf microbiome, the seed microbiome, seem to be very poorly connected to any other type of data. So this gives us some indications about what to expect. So these are results of random forest models that we trained for all of the phenotypes that were measured based on the expression data that we gathered from all of these individual plants in the fall. And so you see the ones that actually are predicted the best are the ones integrated with the green arrows here, which are mostly early leaf and rosette phenotypes, which makes sense because these early leaf and rosette phenotypes are more or less contemporaneous with the RNA sequencing data that we generated in autumn. And as far as the leaf phenotypes are concerned, they're even on the same material. So, but you see that even for some of the final phenotypes, so the ones indicated here in blue, actually most of them, we still get better than random prediction performed. So we can actually, for individual plants, to a certain degree, predict what is going to be the yield of these individual plants based on transcriptome data that was generated 5 months before. By the way, these 95% confidence intervals you see here were pretty difficult to generate. So my postdoc Stan developed a method for this, which was published last year in American Statistician. So if you look at one of the early phenotypes, leaf length, the gene expression data, which is the all genes thing over here, we see that it's pretty predictive of phenotypes. Also, if we limit it to only transcription factors and eliminate all of the other genes, if we only look for transcription factors, it's still more or less the same performance. But all of the other data layers, like the sensors, the microbiomes, the nutrients, actually have predictive performance which is no better than random. So we cannot predict leaf size, for instance, from the nutrient profile at sowing. That's not possible. On the other hand, if we use other early phenotypes to predict one of the early phenotypes, like leaf length, we have almost perfect prediction performance, which is not surprising because all of these are really highly correlated. More surprising is that if we do the same thing on a final phenotype like total seed weight, again, the transcription models do pretty well, but the early phenotypes are equally good predictors of final phenotypes. So from the omics perspective, this is bad news, because why would you generate very expensive omics data to predict what the final phenotype of a plant will be if you can just as well take a picture of the leaf and then predict it from there, and you have the same performance, okay? But on the other hand, these early phenotypes don't really give you any idea about the underlying molecular events that influence the yield phenotypes of the plants, and that's what we use the omics data for. So for instance, what you see here is the top 10 transcription factor predictors for yield phenotypes. So there's a bunch of yield phenotypes here at the bottom in this matrix, which is like blue to yellow. All of them are pretty highly correlated. So shoot, dry weight, total seed count, total seed weight, total branch count, and so on. And then at the top, in the top panel here, you see the expression profiles of these top predictor genes that we got from the random forest model. And you see that there's actually two classes. There's a bunch of genes that is very highly expressed in genes that have low or poor yield. And then you have a bunch of genes that is more highly expressed in genes that have higher yield. And then if we look what kind of genes these are, for instance, for seed weight, the ones in red here are positively correlated with yield. The ones in blue are negatively correlated with yield. And so most of the red ones, so the positive correlates with yield, are involved in the floral transition, multidimensional cell growth, cell differentiation, etc. So indicative of being involved in one of the transitions that takes place and is known to take place actually in Brassica napus in autumn around the time point that we actually profiled the transcriptome of the leaves. And then on the other hand, the plants that do poorly have higher expression of genes which are involved in abiotic and biotic stress. So this seems to suggest that some plants in the field were stressed. As a result, they had a delay in development, that they did not undergo the floral transition, or they were not in the same stage of the floral transition at the same time. And this gave you a lower yield at the end of the season. So it suggests that there's some kind of developmental gradient in the field. And in order to investigate this in more detail, we relied on techniques from the single cell field. So single plant and single cell data are pretty similar in the sense that they're all observational. They're generated on individuals. You can use the same kind of techniques on both kinds of data. So we actually did the diffusion mapping of the transcriptome, then inferred a trajectory from these data and did pseudo-time inference on them. And then you can essentially order your samples along a pseudo-time axis. And when we do this and we plot the pseudo-time of the different plants in the field, we get essentially something that is very similar to the factor 1, so the most important factor explaining variation that we got from the Mephisto analysis. So this makes sense. Pseudo-time captures a lot of variation happening in the field. And we know that this is pretty highly correlated to transcriptomes, but also to phenotypes. So if you plot, for instance, the total seed weight as a function of pseudotime, you see that plants with high pseudotime generally have higher seed weight. Plants with low pseudotime have lower seed weight. So there is some kind of gradient in this field. Okay, the next one will take a bit too low because it's a pretty heavy figure. But what you can do then with pseudotime is, for instance, look at what kind of genes are differentially expressed across pseudotime and what kind of genes are they. And then we can order them, like for instance here. You have genes that are highly expressed in plants or early on in pseudotime. Most of these are involved in abiotic stress and leaf senescence. And then you have a bunch of genes that are kind of peaking at intermediate pseudotime phases, mostly involved in auxin responses, but also photomorphogenesis, indicating that they're kind of on the edge of doing some developmental transitions. And then if you look at the later pseudotime genes, all of these clusters you see here are enriched in genes involved in the vegetative phase change, the floral transition, and leaf maturation geoclasses. So it seems that indeed there's a developmental gradient in the field, and with our single cell techniques, we can actually capture this. And then the question is whether this pseudotime still makes, let's say, sense. So can you look at the gene expression profile across pseudotime and conclude anything from this? And so we looked at the, where is it? Oh, here. One of the most important regulators of the floral transition, FLC. So these 9 here are Brassicanaphus orthologs of the Arabidopsis FLC gene. There's 9 of them in our data set. And then if you look at them, they all kind of show different kinds of profiles. Some of them are not significantly differentially expressed across pseudotime. Some of the others are. And the ones that go down most strongly, which is what you would expect for FLC genes during the floral transition, these 2 here are exactly the same ones as the ones identified by 2 other studies as decreasing the most fast upon cold treatment, so during vernalization. So this kind of indicates to us that our pseudotime profiles make sense. The ones, these 2 here in red, that are not moving at all, it seems, these are the ones that are generally believed to actually determine the strong vernalization requirement for winter type Brassicanaphus. So winter type rapeseed has a strong vernalization requirement. These 2 genes are supposed to be the most important ones. It's also natural that then over here in the early stages of the floral transition, they don't move. They stay high. They should only fall lower later on in the floral transition. And then the other ones over here, we see some weird stuff, like for instance, things going down and going up again. This has also been observed upon cold treatments for these genes also in different studies. So this tells us that the profiles that we capture from our single plant data ordered along pseudotime using computational techniques can actually more or less mimic time series that you would classically generate. And so this then also opens perspectives for linking the different data layers together and maybe even doing causal inference on them. So this is something that we're investigating right now. So we can already link the transcriptome layers here with the phenotypes. I've also shown you that the other data layers, like for instance, the leaf microbiome, are not directly predictive of any phenotypes, but we can link them actually to some of the early data layers. So we can link leaf microbiome or nutrients to the expression of particular genes in the field. And then these genes are linked to certain phenotypes as well. So we can indirectly also start linking nutrients and microbiome components to phenotypes. But this is still essentially work in progress. So take-home messages. I hope I've convinced you that by profiling phenotypes and microenvironmental variability in a field setting, that single plant omics, the technique that we're advocating, can actually uncover what kind of processes are active in a field setting and how they influence the phenotypes of the plants. I want to stress that single plant omics is conceptually similar to single cell omics, so we can use the same kinds of techniques, which is an advantage for us because there's already a lot of techniques developed in the single cell field. But whereas single cell data focuses on cell differentiation and cellular phenotypes, with single plant data you can really go to more global plant development, stress response pathways, and analysis of macroscopic phenotypes like yield. And so in conclusion, we think the single plant level has erroneously been skipped when people were transitioning from traditional systems biology on pooled plant samples to single cell studies. So thank you very much. And thanks to these people that did most of the work, also from the labs of Hilde Nielsen and Dirk Zee, from Sophie Goormachtig, who helped us with the microbiome analysis, from Isabelle Roldan, who helped us with the field trials, or what we call the field trials, and then also funding sources, obviously, in particular the FWO for granting us some money for SAM, Syngenta for paying for the MACE data generation, and Inari for paying Steen Hawinkels' fee. Thank you very much.
Thank you, Stephen, for that excellent presentation. We have maybe time for one question. Anyone in the audience who would like to ask a question, please come on. Yep.
Thank you very much for the great talk. So I actually work in the field of spatial transcriptomic data. And so you made a point about the similarity with single cell data. And I'm wondering if you see that there are any challenges in using techniques that are specifically developed for spatial transcriptomic data and using that in plant data. And conversely, do you see that there are tools used in plant data, in single plant data, that could be used in spatial transcriptomics that are not being used?
Well, the setup is maybe a little bit different in the sense that our plants are just positioned on a grid. Mm-hmm. This makes it for us a lot easier to take into account the spatial effects. But indeed, there's a bunch of spatial effect correction algorithms, which I think are underused in spatial transcriptomics currently, especially if it goes into like hypothesis tests and so on. You have to really take those effects into account. This is not always done correctly. And there's a whole array of different techniques. Steen, actually, my postdoc, who is a statistician, did a review on how these methods, how well these methods work, and he found that some of them work well, other ones do not work at all. So you have to be really careful which ones to apply. Okay. Something else that Steen, for instance, is working on right now. So he's actually transitioning into the spatial transcriptomics field. There's a lack of statistical methods to do hypothesis testing if you're working with point patterns. And so the spatial transcriptomics technologies where you actually get individual molecule localizations, there's a lot of technology development still to be done there.
Okay, thank you.